In this video, I introduce the concept of yielding elasticity, including concepts and definitions and ideas about the underlying structures that cause this behavior. Yielding fibers are any fiber that undergoes large reversible deformations at constant stress. So if we show a stress-strain curve, while initially the stress increases, at some point it undergoes a large deformation at that constant stress. So this occurs when the fibers undergo some sort of a reversible phase transition. A common cause of yielding in biological materials is unfolding of the proteins inside the fiber. As long as the proteins are covalently bound together or otherwise bound in some way that they maintain contact with each other even when they unfold, that is, they don't unbind when they unfold, they can exhibit this yielding elasticity. So we show here a picture that shows these several phases. There's this elastic regime alpha where the individual fibers stretch without breaking the structure of the protein. So this is a picture here, an example of alpha helices that make up spectrin in the red blood cell membrane. So we see here there's this alpha helix structure that can stretch. And then there's this next regime, this unfolding or yielding regime, where this undergoes a phase transition. I call this more generally yielding because they don't always happen because of unfolding. But we see here that these start to unfold and then they'll continue to unfold over during this regime. And once they're fully unfolded and they look like we show here in gamma, then they can still stretch due to covalent stretching of the bonds that make this up. But at that point, they'll be much stiffer. And so they exhibit then this strain hardening behavior until stress gets so high that this fails. So this creates what we call an S-shaped force extension curve. It's actually kind of a backwards S, but it's still called that. And this is due to these three regimes. So the functional advantage of this is that this elongation protects the material from the high stress that will cause it to fail. So to consider this, think about a paper. If you've ever tried this, if you tear a little piece of paper like we show in this picture, and then you pull across the tear along the flat way, what you'll see is the paper will deform and all of the stress that you're applying this force, but it really gets concentrated at the edge of the crack where the stress gets very high. And we refer to this as stress concentration. What's happening here is that the strain is highest at the edge of the crack. And because a linear material has the stress proportional to the strain, the stress is also highest there. And in this situation, the rest of the material really isn't helping much with the load. It has very little strain and thus very little stress. And so all of the force gets concentrated there until the stress at that crack exceeds the ultimate stress and the material fails. When that happens, the crack grows, and now the new edge of the crack has all the stress concentrated on it. And so this cycle is called crack propagation and leads to complete failure of the object. The amazing thing about yielding elastomers is that they disperse the stress. So because the stress is equal to the yielding stress, independent of the strain within that phase transition region, then you can have large parts of the material that are strained to different extents that can help equally with the load. And this can prevent the stress concentration and the crack propagation. The image we show here are molecular dynamic simulations performed by Marcus Buehler's group, and the Akbara reference from the previous slide. And they're showing here in yellow all of the fibers that are in that yielding regime, whereas those blue ones that are mostly across, in this case they're pulling the fiber uniformly across the top in this position. So the fibers moving this way have no stress on them. They're still in that linear range, as well as the fibers that are here in this region where around the crack or the tear in the material, because the material is deforming, they're really not under stress. But all the rest of the material you can see is yellow and is in this area of equal stress, even if they may have very different deformations. And so this large deformation these can undergo protects the material from this stress concentration and crack propagation. So while these materials are yielding fibers and materials are almost invariably also strain hardening. And that is because they're stiff in this low force region, and then they become very soft in this yielding region. Once they have fully undergone the phase transition, they'll be stiffer, they become stiffer again. And so that is referred to as strain hardening regime. And in fact, a lot of polymers that undergo phase transitions, because they're unfolding, or simply because polymers often act that way, they're almost always worm-like chains that undergo these strain hardening behaviors, and the stress goes up. So a material composed of these yielding fibers are generally strain hardening as well as yielding. And we show here a stress strain curve where we show the yielding region and then the strain hardening region. Again, the S-shaped curve can reflect the fact that the fibers are S-shaped, that the fibers can rearrange, adding to the strain hardening, or multiple types of fibers. That is, all of the things that cause strain hardening in materials can also cause the strain hardening in materials made up of yielding fibers. The only difference is this at very low stress, that there may be a region where they're stiffer prior to the yielding. A lot of times this yielding behavior may barely be visible at the material level, depending on the sensitivity of equipment. But even so, the ability of these fibers to yield is still protecting the materials from stress concentration at the molecular level and preventing tearing. For example, these spectra networks that make up red blood cells undergo very large deformations while red blood cells squeeze through blood vessels, and it's thought the yielding behavior is part of what protects our red blood cells. In addition to the spectrum of red blood cells, there's many other examples of yielding elastomeric fibers in biology. So anything that's shaped like a helix generally has yielding elasticity, although the degree, the length of that yielding phase will depend on how much the fiber elongates when it undoes the helix. So a DNA double helix is a fairly shallow helix like this, or a fairly long helix that only elongates a little bit at the phase transition, but it does undergo one. There's other filaments besides spectrin, other types of mostly intermediate filaments that are alpha helical. And then there's also quaternary protein helices. There's a number of bacterial fimbria, including some that my group studies, that fold globular proteins up into a helical structure that can be extended under force, creating the same types of yielding elasticity. In addition to helical proteins, any multi-domain protein that can unfold, and I say multi-domain, I guess it could be multi-domain or multiple subunits covalently bonded together. It just is, as I mentioned before, requires a mechanism whereby unfolded proteins don't lose their ability to bind to each other, which usually requires their tertiary fold. So one example of this is titin molecules, that these are in muscle. They have an amazing approximately 100 amino domains that all can unfold in addition to some other domains. And these form a structural basis for our muscles that can help retract and allow the muscles to contract if they've been overstretched. Another example is fibrin in blood clots contain molecules that unfold, and that may protect the blood clots again from tearing. Finally, we won't talk about this as much in this class, but any semicrystalline material with subunits that can change shape can be yielding. In this case, there may not be unfolding the same way, and they may not elongate as far, but they can still undergo phase transition. Examples are lipid membranes, And we'll talk about this when we talk about cell adhesion next week. Or the one biological example is something called shape memory alloys, which are crystalline materials that have two different crystal shapes that have different sizes, that have a different crystalline packing, causing them to elongate in one direction and shorten in another. And these are often used to earthquake protect buildings because they absorb a lot of movement and energy as they change shape back and forth. Almost all of these examples of phase transitions exhibit hysteresis, that is, yielding materials are viscoelastic. And we see an example here that we've shown here. If you pull Titan molecules slowly, you can actually see the sawtooth pattern as each domain pops open one at a time. And this is a short region of just 8 domains that are pulled. Now, if in fact you pull a full Titan molecule of hundreds of domains, and you pull more slowly, these even out. That the softness of the long pull along with the slower pull causes those sawtooths to even out. And what you see then is you still see as you stretch the initial, and then as you undergo the phase transition, we see that yielding behavior there. And then when you relax it, it goes back on a different curve at lower force. That's the hysteresis in here. But if you go slow enough here, you can actually see these start falling on top of each other. And so you lose that hysteresis. Now, these viscoelastic materials cannot be modeled with the linear methods of week 7 precisely because they're nonlinear. This is a nonlinear behavior. Even at equilibrium, these are nonlinear behaviors, so we can't use the linear models. In summary, yielding elasticity is caused by phase transition, such as protein unfolding, and serves to protect materials from stress concentration and crack propagation.